Hi everyone. So our next topic is Taxol. Okay. So our next topic is Taxol. So already we have discussed about Taxol in our class. That is a pharmacognosy of Taxol. So now we will be dealing with the estimation, isolation and utilization of Taxol. Okay. So firstly, Taxol, biological source. So it is obtained from the bark of Taxus brevifolia belonging to the family Taxaceae. Okay, so it is obtained from the bark of the plant name is Taxus brevifolia belonging to the family Taxaceae. Also found in various other parts of various other species of taxus like Taxus baccata, Taxus cuspidata, Taxus canadensis, etc. Okay, so it is also seen in various other species of taxus and in various other parts like leaves, flowers, fruits. Okay, so in those parts from those species, Taxol can also be obtained. Next, so coming to the isolation part. So we generally have two methods in this. Coming to the first method, powdered bark is been taken and it is extracted with ethanol or methanol. Okay, so powdered bark is been taken and it is extracted with ethanol or methanol and filtered. After the filtration is done, it is evaporated at 40 degrees centigrade. After the filtration is done, it is evaporated at 40 degrees centigrade and dried residue is obtained. When you dry it, when you evaporate it, you will get a dried residue. This dried residue is dissolved in alcohol. Okay, it is dissolved in alcohol and filtered. You will get an alcoholic extract, right? That is again evaporated and again you will get a dry residue, right? To the dry residue, okay, carbon tetrachloride and water is added. To that carbon tetrachloride and water mixture, okay, is been added, and when you centrifuge it, you will get two layers. Then centrifugation is been done, then you will get two layers. Okay, in that you will take the carbon tetrachloride layer. Okay, this centrifugation or this what we say, yes, after adding this CCl4 and water, this thing has been repeated so many times. Okay, this whole process has been repeated. These two steps are there, no? These are repeated so many times so that you will get carbon tetrachloride layer so many times, and all those carbon tetrachloride layers are been pulled up and they are collected together. Okay, and then the whole thing is been evaporated and dried. Okay, and again they are been dissolved in carbon tetrachloride and methanol 1:1 ratio. Okay, they are dried, they are dissolved again in carbon tetrachloride is to methanol 1:1 ratio. Okay, and then again they are filtered and evaporated. Now it's purified by chromatography, by preparative TLC or by column chromatography. So again the column is been kept, and when the column is been run, you will get crystals of Taxol. Okay, that is what we do. In the second method, it is somewhat similar to that of the method 1. So the powdered drug is been taken and it is extracted for 16 hours with methanol. So firstly it is extracted for 16 hours with methanol. Then it is filtered and concentrated and dried at 40 degrees centigrade. And again the same process is repeated. That is, again you add the methanol and you extract for 16 hours. And then again the filtration is been done and concentrated and dried at 40 degrees. And then the residue is extracted with dichloromethane and water. Okay, after the whole thing is repeated and you get an extract, that residue has been extracted with dichloromethane and water, and it has been filtered and centrifugation has been done. Okay, and then you take out the dichloromethane layer, then it has been evaporated to dryness, that is been treated with methanol is to dichloromethane 1:1 ratio. Then you get the crystals of Taxol. Okay. Next, there are some other methods for the estimation of Taxol, for the isolation of Taxol, like supercritical fluid extraction. Okay, that is one method, and you also have a plant tissue culture method. So that is not like, that will be very lengthy and it is not that much essential to know. So these are the two main methods which you can write down, and if 5 marks question is given, you can write down only one method. That is enough. Next, estimation process, TLC. That is stationary phase, silica gel G is been taken. This is common, right? So silica gel G is been taken. Next, melting point, chloroform is to methanol. Chloroform is to methanol, 7:3 ratio is been taken. Next, detection, 1% vanillin sulfuric acid is been sprayed on the TLC plates. 1% vanillin sulfuric acid. Okay. In the later case where artemisinin was taken, we have taken NS-aldehyde sulfuric acid, right? We have taken NS-aldehyde sulfuric acid. Here we have taken vanillin sulfuric acid. Okay. Next, the TLC plates are heated at 110 degrees centigrade for 10 minutes. Blue color spots are been obtained, and after 24 hours, these blue color spots are converted into dark gray color. Okay. And the RF value is like 0.90. That is about the estimation. That is first one, TLC. Next, when we take up the next one is HPTLC. Okay, when we go for HPTLC, the stationary phase is silica gel GF254. Melting point is chloroform is to methanol, same like this, but here 7:3, here 9:1. Okay, sample 1 mg per 10 ml in methanol. So 1 mg in 10 ml of methanol is been taken as the mobile phase, as the sample. This is how we prepare the sample. And the standard various concentrations starting from that 1 mg or 1 μg to 1 mg, that much you can go with various concentrations. Okay, and the detecting agent is vanillin sulfuric acid. So same thing, we place the on the TLC plates, we keep the spots of various concentrations of the standard, and the sample concentration we already know. So those things have been kept, and where we get the spots, similar spots, those samples are been taken out, and then again they are been kept in the densitometer at 235 nanometers at 110 degrees for by heating the plates at 110 degrees centigrade for 10 minutes. Then you get beautiful peaks. Okay, these curves, calibration curves are been compared with each other. That is about the test and the sample, and you will get the exact RF values of that sample and the standard. Okay, next coming to the last one, that is the HPLC method. Here the stationary phase is reverse phase column 18, and the melting point, just remember one thing, you have all the things in general, common things. HPTLC ki silica gel GF254, HPLC ki reverse phase column 18, alantivi konni meeru complete ga by heart gurtunchukochu. Okay, next stationary phase, as already discussed, melting point, methanol is to water. Okay, here all things we have studied, chloroform is to methanol, chloroform is to methanol. Here methanol is to water, 80:20. Okay, flow rate should be, here we do the injection, right? Ejector is present, okay, which injects the sample in the stationary phase. Sorry, sample in the standard. So here flow rate is 10 ml of sample with flow rate of 1 ml per minute. Okay, so that is the flow rate, and UV wavelength is at 227 nanometers. So finally you get the retention time when you compare the peaks with that of the stationary as well as the the sample and the standard. You get the retention time of 2.05. That is about the HPLC method. And finally, utilizations. It mainly acts as an anti-neoplastic agent, that is the anti-cancer agent, and mainly it is helpful in lung, cervical, breast, prostate, colon cancers, etc. And also it acts as a anti-proliferative agent. Okay, so these are some of the utilizations of Taxol. This is the Taxol topic.